Hello, my name is H Wang, and I'm a neurologist and researcher who takes care of and studies patients with muscle and nerve diseases at the University of Washington. My goal is to talk about facioscapulohumeral dystrophy, or FSHD. In this video, I'll be talking about the molecular biology of FSHD, and specifically about its genetics. First, we will be exploring how genetic information, such as DNA, gets converted to RNA and then to protein, and then causes trouble in muscle. Let's first understand the concept of DNA. DNA is the genetic repository of all that we inherit from our biological parents. It is coded by 4 letters, ACTG, and this code is in a long strand up to 250 million base pairs, 250 million letters. These long strands of letters get combined into what we call the chromosome. These chromosomes, here depicted by the Encyclopedia Britannica at the bottom, is usually consisted of a complex of proteins and DNA that is called chromatin. This is how we store DNA most of the time. When we don't use parts of the chromosome and parts of genes, they're compacted down into this chromatin and packed away. In humans, we have 23 chromosomes, 22 autosomal chromosomes, plus 2 sex chromosomes. Patients with FSHD have changes to chromosome 4. At the tip of this chromosome 4 is an area where we call 4q35 on the long arm of chromosome 4. In this area, we have what we call D4Z4 units. Normally, we have 11 to 100 of these units, and these are usually just stored and do not cause any problems, nor are they being used. Think of each of these repeat units as a storage unit. Somehow in our evolution, we have multiple storage units repeats that are stored in an industrial warehouse area of the same size storage unit. For the most part, they are compacted down in the chromatin as part of the chromosome, and they are packed tightly and not being used in adults. However, in certain cases, there can be fewer of these repeats, and when you have 1 to 10 copies, the storage units become more accessible, and what is stored in each storage unit can be accessed. And in those cases, that gene that can be turned on is called DUX4. DUX4 is a transcription factor, meaning that it encodes a protein that can in turn turn on other genes. For the most part in adulthood, DUX4 is quieted. It may have a role in development, meaning that it may be important when we are a fetus, but gets turned off when we become adults. So this is what happens in that the DUX4 gene gets turned on, it gets expressed into the DUX4 RNA, and then the DUX4 protein, which is a transcription factor. And this transcription factor is what wreaks havoc in muscle. Let's take a grander view of DUX4 and see how it causes disease. Here, the DUX4 mRNA is the center of what causes disease in FSHD. Normally, we have more than 11 of these storage units, and they get methylated and condensed they get stored away. In such cases, there's no FSHD. In FSHD1, what happens is that there are 1 to 10 repeats. It's a smaller repeat, and combined with what is called a permissive haplotype, DUX4 is able to be accessed in the storage units and be expressed into the mRNA. There are cases that we call FSHD2, and this affects about 5% of patients with FSHD. In these cases, there are about 11 of these D4Z4 repeats, but somehow they don't get condensed, they don't get packed in and stored away in this industrial warehouse place. And therefore, each of these D4Z4 units could be accessed, and that causes the DUX4 mRNA to be transcribed. We have found 2 genetic mutations that can cause all of these storage units to be more accessible, to be more open, and for the chromatin to not be as condensed in this area. These 2 genes are SCHMD1 as well as DMMT3B. Mutations in these genes can cause a more open heterochromatin, and the accessibility of the DUX4 DNA is more available, allowing for the DUX4 mRNA to be expressed. So what is important is what we talked about in terms of in patients with FSHD, the DUX4 gene is more available in adulthood when it shouldn't be, and it gets inappropriately accessed, inappropriately expressed to the DUX4 RNA and then to the DUX4 protein, and this is what causes disease. Let's focus a little bit on the DUX4 protein and how it may cause problems. Here is a slide of muscle cells in a petri dish. Here, the researchers have taken muscle cells, put it on a slide, and tried to look for DUX4 activity. DUX4 activity here is measured in the redness. And each blue circle is the nucleus of the muscle, and muscle cells tend to have multiple nucleus. You can see on this slide that there are multiple nuclei, but not all of them express DUX4. And there are certain cases, randomly in certain nuclei, the DUX4 is being expressed. And this turning on DUX4 seems to turn on this whole slew of proteins that lead on to a program that causes the cell to become sick and to die. So what is another way of looking at this is that this right here is the muscle of a patient with FSHD. Here, they're really not affected, and you can see that each cell is pretty healthy. They're rounded, and each nuclei are on the side, and each cell and muscle fiber is connected to each other. In a patient with FSHD, DUX4 gets turned on, and this is the transcription factor. This transcription factor in turns on new genes, and these genes that get turned on causes the turning on of all these programs that lead to the pathology of muscle that we see when we look in the muscle of a more severely affected patient. Here you can see that each muscle fiber, each cell is of different sizes as opposed to when it was this uniform size. There is some degradation of the muscle fibers, and you have these purple cells on the right of the slide that are coming in to try to clean up. So this seems to be what happens in patients' muscles with FSHD. There is an inappropriate activation of this gene called DUX4, and this in turn turns on a slew of proteins that lead to the degradation of these muscles. And this seems to be what affects our patients with FSHD. This video is brought to you by the University of Washington Paul D. Wellstone Muscular Dystrophy Cooperative Research Center in collaboration with the Friends of FSH Research, for which we are grateful for their help and collaboration in our research projects. Thank you.